Hi everyone, my name is Lindsay, and today we're going to be going through the features of Watson and Crick's DNA structure. DNA molecules are made up of subunits called nucleotides, and these nucleotides are arranged in a double helix structure. Nucleotides are the building blocks of DNA, and they are composed of three parts: a sugar, a nitrogenous base, and a phosphate group. First, let's look at the 5-carbon sugar as shown here. We label the ribose carbons 1 to 5, starting with the carbon 1 attached to the nitrogenous base. Then we move to the next door carbon and continue labeling. The 2' carbon distinguishes DNA from RNA. DNA has an H while RNA has an OH group. The 5' carbon is attached to the phosphate group, and the 3' carbon bonds to the phosphate group on the next nucleotide. These phosphate groups form the sugar-phosphate backbone, and they are held together by phosphodiester bonds. Next are the nitrogen bases, and there are 4 different kinds: adenine, guanine, cytosine, and thymine. A and T always bind together, and C and G always bind together. A and G are purines with double ring structures, while C and T are pyrimidines with single ring structures. The forces that hold these pairs together are called hydrogen bonds, and they are relatively weak. To help you remember these base pairs, let's put them in terms of food. We'll think of adenine as spaghetti, thymine as meatballs, guanine as waffles, and cytosine as chicken. When you pair these bases together correctly, you get spaghetti and meatballs and chicken and waffles. If you mix up the pairs to make spaghetti and chicken or waffles and meatballs, your food probably won't taste so good. As previously mentioned, the hydrogen bonds holding the base pairs together are weak, and they are not the main force stabilizing the double helix. Instead, there are strong hydrophobic interactions amongst the base pairs that work to keep the DNA strands together. These interactions are maximized via base stacking, for the double helix is stabilized when the flat base pairs stack on top of one another. A key aspect of DNA is the direction of the strands. Each strand has a 3' end and a 5' end, and this is determined by the direction in which the nucleotides are connected. One strand is oriented in the 3 to 5 direction, while the other is in the 5 to 3 direction, and this means that they are anti-parallel. DNA strands can also vary among the direction of the helix. Right-handed DNA has the helix pointing up to the right, and left-handed DNA points up to the left, as shown by these 2 spiral examples. Almost all DNA is right-handed, but there are a few cases where left-handed strands are present. As you can see here, the grooves within the helix are not evenly spaced. They alternate between wide and narrow section called the major and minor grooves, respectively. Because of this feature, more proteins are able to interact with the base pairs in the major grooves compared to the tighter minor grooves. Finally, let's briefly discuss DNA melting properties. The phosphate backbone we talked about earlier has a negative charge, which acts as a destabilizing force within the molecule. If we added some DNA to a salt solution buffer, however, the negative charge would be partially neutralized, reducing the destabilizing forces. Thus, the DNA would be held together more tightly, so the amount of heat required to denature or break apart the molecule would increase. We call the point at which 50% of the DNA is denatured the TM, so using a salt solution buffer would ultimately increase the DNA's TM. Here's a list of my sources, and that concludes this video on DNA structure. Thanks for watching.